Yeah. Um, in a sense, I mean, the reason why we are having these discussions is because we're trying to put together a research proposal gathering collaborative partnerships. And in UK, FAPESP has um, a special program with BBSRC. The deadline is 4th of October. And with Norway, Bjorn, there's really a lot of new stuff being developed. And I just made a whole list of different things. And in the US, we're talking about the funding agency of the state of Sao Paulo. We've and our Minister of Science and Technology has been in Norway, I think 2 months ago, in a discussion. And in fact, I mean, Norway was a little bit disappointed that Brazil has not managed to save as much of the rainforest as they promised to do. And then they cut some of the $1 billion that Norway is giving to Brazil. I think in 2010, 2011, Norway allocated $1 billion. And this money is not being fully used, the money on there are possibilities. And fast dates and right. First of August. Oh no, but it's the idea that we don't need. I saw him. First of all, let's wait. Oh, really? Okay. Okay. Yeah. Yeah. Very well. First of all, I think we should probably start talking and let's see. We have people from Uruguay coming, Ungerfeld, and oh, he's the local. He's here. Yeah, yeah, and he's going to be joining us. It's probably a little bit late, and my only concern is that it's not an easy place to find. No, it's not. Well, but this morning when I'm trying to find the interview, I looked. I tried to look it up and find it, and I went to the building, and this is the first two people. No. That's a bit of a concern. But in a sense, I mean, what I feel is that a little bit like redoing what we did with the AWIN back in Oscar board, with the work package 3 type of Pre-Natal programming. Yeah. The thing now that the reason why it's quite an exciting thing that because he's now working on epigenetic toolbox, and in fact they just published something quite exciting in terms of making this technique a bit more. You're writing it, Fabio. Yeah, Fabio. Yeah, Fabio. Yeah. And the first question I was going to ask you was how much is this observatory about epigenetics? a bit of a mixture, a lot, because the way that and I kind of made the decision that we're going to make that use of that very little. The issue is kind of discussion and going to the board, but the real issue for me here is, do we have enough protocols to fully understand the consequences of a, a pregnancy that went well, not so well? I mean, are the protocols available good enough? I mean, back, let's say Patricia in her works now using open field test, novel object test. I mean, is the behavioral toolbox what we need? I mean, and then we're merging now with another scientific discipline like epigenetics, and that's basically what I expect the workshop to be about. This kind of putting together the different areas, and I want to and for me what is really exciting the possibility to make this comparison a lot of challenge with some of the viral diseases like dengue, chikungunya, Zika, that the severe consequences for the offspring. And in a sense, I mean, the running together these type of areas, and we have, I mean, one of the that I co-supervise the PhD novel using LPS that produce kind of a inflammatory response during pregnancy, and we found some differences in the offspring. And then, I mean, the thought is basically kind of getting back to this discussion and see how much of this type of area is an area that we can build a comparative type of scenario. And for comparative work, funding is not an issue, but if you have a kind of a human element of that, and in the end, we put something quite practical that has to do with the opportunities for grant proposals, and then we have specifically this 4th of October timeline. Already, Fernando emailed me now saying that he's keen in collaborating. We can add something on pathology, and that was the kind of thing that we put into the program. And I put two different questions, and if we provide, I kind of said it again, but basically, if we really are happy with the available protocol, the methodology to assess the consequences of the prenatal environment, and if given the how broad is the area, if there is a way of us creating some opportunity for collaborative research. Getting to very practical issues, the prenatal and early postnatal mortality, it's incredibly serious in South America. Don and Bjorn, I discussed with lambs. The industry loses over 30% of lambs, and that's the subject of study of Rodolfo Ungerfeld that's coming here. It's quite
everywhere. But lamb mortality is high.
Very in South America.
I mean, compared with when you think about how well developed the animal is, and yet we have a lot of mortality.
Yeah.
I mean, obviously, the more nutritional the species, this is a very well developed.
Right.
And it's a very high mortality.
Very high mortality.
To do, well, I would say it's really to do with how the animals are being kept in conditions where nobody is giving them enough time. And maybe not just that.
No, and
People don't put much time into looking after them. But what they do, and what they do is in a very short time. So if there is care of land around there, that's a very short time. But if there are other factors,
yeah, and then this is and we at the moment one of the things that we are kind of getting interested is also the potential role of the males. I mean, because we kind of the female germline kind of gets organized during pregnancy, but the male germline is kind of gets replaced over and over again. I mean, then we have now two projects where we're looking at potential impact on the male germline, whether or not there's been a potential contribution for the phenotype on the base of that. And one of the, and I can talk a bit more, but we with this prenatal injection of LPS that reduce fever in the female in the last third of pregnancy, what Fabio found was that the testes of the male got smaller, got affected by being exposed as a fetus to this environment, and also having some results that showing that not these fibers, another fiber that shows that the ability of the lungs to make choices becomes kind of compromised as a result of this exposure. But males seems to be affected. He's done a lot of work later on looking at male choice and sperm quality that is not being analyzed. And that's the kind of the background. Maybe would you like to introduce? Why don't you introduce? Yeah. 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 Why don't you introduce yourself? Then I'm going to go. Well, I'm in the video.
Yeah.
Or should I organize? You can talk about yourself. When should we can find someone else?
My name is Patricia. I'm a PhD student. I'm a biologist. And now in PhD, I am trying to understand the effects of environmental enrichment during gestation, the outcomes in the offspring. So I'm working with swine. So we put straw to the sows during gestation, and we have some interesting outcomes in the phenotype with abnormal behavior in the piglets when they are from the sows with straw. So the piglets that is from the sows without straw, they have stereotypic behavior more than stereotypic behavior, the nosing. Yeah, each other inside the pen. Yeah, it's a kind of belly nosing, but I saw just sometimes the belly nosing, typical belly nosing, but the another times I saw in another parts of the body. And I had more interesting results with the sows that express stereotypic behavior. So the piglets from sows that has high stereotypic behavior express less fear in the emotional tests, fear tests. And we're trying to understand this. I had some physiological outcomes too with saliva, cortisol saliva in the saliva, and it's a little bit mess because we can't. It doesn't make sense. Yeah, with saliva cortisol, and we did some analysis in the placenta with cortisol and cortisone, and and we have the outcomes that cells that express more stereotypic behavior, the cortisol is higher, the placenta. Just this, but when we see with environmental enrichment, there is no difference in this concentration. And now I will do in Linköping University the epigenome of the brain in these piglets. we have some tissues that is cortex, amygdala, and hippocampus. And we will see the epigenome there in these tissues. Because we have some difference in the phenotype, so we...
How are you going to get that information?
Which one?
How will you find that information in the epigenome? What are you finding? What are you measuring?
We will do some protocol there that Fabio can explain better because he developed one. And we will see the methylation, the sites of methylation.
So methylation sites of
It's a group of things, right?
Yeah, it's like you do the whole methylation analysis or do you reduce it? That one that they...
I don't know so far.
Okay.
I think
So
that is
then
whole
we,
methylation.
Yeah, I will talk a little bit what we did and I will explain better to understand. But it basically is like you have both conditions and you sequence the genome of the animals in both conditions, but you use antibody that you to do the immunoprecipitation of methylated sites. So as soon as you have the DNA, the immunoprecipitation, you can isolate the fragment that is methylated or that is enriched with methylation, and you sequence both treatments. And after this, you sequence both treatments and you check the coverage, because if you have the sequence, you sequence this.
You are finding how much information there is in total.
Yeah, you will find different coverage between the treatments. So with the bioinformatic analysis, you can measure this and you can say, okay, this region is more methylated in this condition and less methylated in this condition. And you compare all the...
You are doing it reason by reason, not that perfect.
Usually people do with the whole genome, but we developed a method to reduce the genome. But we did not develop this method. This method comes from Cornell. Yeah, they developed a method to reduce the genome. So they use some specific enzyme. So the enzyme cuts the DNA, and according to the distance between the sites that the enzyme recognize, you have a different fragment length. So after you digest your DNA, you select the appropriate enzyme to enrich fragments with more or less 200 and 500 base pairs that is suitable to be sequenced. Okay, and so you can sequence just that fragment that has that range that you want. So you reduce the genome. I don't know if I can show some slides and
maybe when people arrive,
yeah.
The reason why this protocol is kind of new, I think the techniques in the past, I think they create the bias when you give this type of.
Because usually when people want to reduce the genome in this methylation analysis, they use the enzyme that cut the motif CCPGG. And we know that CPG are the nucleotides that is possible to be methylated, right? So what they do is we have some specific enzymes that cut exactly CCPGG. And it's known that the most part of the CCPGG are located in CPG islands. And CPG islands are really conserved. Yeah. So why you look for regions that is really conserved if you wanna look for difference, for change? So is biased because you go directly to the strategy island, and with our method, using we discovered one enzyme that enrich CPG, but the enzyme has no the CCPDG, nothing related with CCPDG. Okay, we don't know exactly why we are enriching CPD islands, but we are. Okay, we discovered that because when we did a study with the chickens, I work with the chickens. Sorry, I didn't present myself because we started in the question, but I will. I will. We discovered that this enzyme enriches microchromosomes in chickens, and microchromosomes in chickens are really rich in genital regions and CPD regions. So
Yeah, I don't know that microchromosomes, these are outside the normal chromosomes, or these
yeah,
are small chromosomes?
it's
in the
small, yeah, it's small chromosomes, and usually in the conventional sequencing, we cannot sequence this chromosomes, or we can sequence some of them, but not all.
Yeah.
Okay, so the thing is, this enzyme cuts the fragments in microchromosomes that is enriched with the CPG.
Yeah.
Okay. So you ask me, okay, but how will you apply this in another species? I have a graph I will show you for that we did in silico digestion to compare the enrichment of fragments that have at least one CPG in different species, like Canis familiaris, Mus musculus, in human, and the standard of fragments is similar. We already applied this methodology to chickens, to in human, we are trying to in one experiment with the people from Linköping, and in Canis familiaris, we are trying to, yes, yes. And yes, in human, the idea is I will try to apply now. After the Congress, I'm going to Sweden, so I will try to apply there in human.
And in pig, I mean,
So,
but this is going to be.
yeah, I have to do in silico digestion to check in pigs how it works, more or less. But looks like the similar pattern of the digestion using this enzyme.
So just the word about this, all this kind of work. It seems to me that you are looking at enormous number of genes, enormous number of areas, and as you said yourself, you concentrate on the ones which are the ones which are most likely to show something.
Yeah,
it's very crude. It doesn't mean it's not going to give any information. It's a long way away from understanding what actually is happening.
Yeah, the idea.
When I read about this kind of area, I feel, well, something is being demonstrated here, but I don't know what is being demonstrated.
But that's the reason why we set up in the workshop, and I hope people develop, they could say the same thing about us doing open field tests or doing cognitive bias tests or doing like she's done like novel object tests, like you did whatever open elevator plasmids. I mean, this whole issue, and in a sense, I mean, you show something, but my what exactly are we trying to answer? And to me, the question about of the prenatal environment is our first choice of get the offspring robust, resilient, and ready. Then I mean, it's a kind of a mistake that we don't want to do. Then before you're right, I mean, Bjorn in 98 wrote about it. You've been saying that for a long time, and things keep moving, and in general, pregnant animals do not quite get this special type of attention. They will basically keep doing things to pregnant animals. And I mean, but my argument, and I think by the end of the afternoon, I hope we're going to have some type of different ideas. I think when it comes to the protocols, I call kind of toolbox to ask the question whether or not the changes did occur, and whether or not, I mean, that's a question we had a lot of, let's say with Patricia, when you see a change in behavior as a result of the prenatal environment, the question is that change always bad? I mean, how can you really make an assessment of if that type of exactly that, see, that's and I feel that at the moment with the
But
type
it's
of
interesting that there is.
correct.
Yeah, exactly.
And would you like to say something?
Ja, because it's the known studies and interesting. Is it possible to relate these changes to any effect on receptor protein synthesis?
Yeah, the thing is, we will do like the idea of the reduction of the genome is to get a good representation of the whole genome. So you have a gene of everything, you know. So as soon as you do your experiment and you do a treatment, you can sequence that, and you can looking for genes that is differential methylated uh, in this condition that in some condition that you want, and you look for the genes. After you have the sequence, you can enrich you can look for pathways. Okay, you enrich the genes to look for different pathways, and if you found something relate with your question, you can go straight to the gene and sequence the whole gene. But the idea is reducing the genome. I can reduce the cost. If you sequence the whole genome of a chicken, for example, the cost will be $50 per sample, right? No, $50 per sample. No, is
The numbers are nothing. You must be
yeah.
right. You can be more focused. You have a feasibility
Yeah.
to do it. But
Yeah, so you can
you can't do it.
yeah, you can sequence it like 50 samples
Yeah.
against one, you know, but they reduce it. So you have the biggest, if you have the biggest sample, it's better to do like a genome-wide association analysis with the trait that you want to look for or something.
Yeah, that's,
Yeah.
that's a different way of putting down there, but also different work the creation of the solar receptors and the countries. This one we talked about 15
Yes.
years ago.
Mm-hmm.
And of course, effect on regulation of the soils, and thereby also the CRH is a fundamental mechanism to find a large number of biological effects. On at least the express, but of course it will be other effects outside that also.
I don't know if you have time to introduce. Why don't we introduce ourselves? I mean, at Raul Zanella, I'm probably the only one who knows everybody from the University of São Paulo. Did you say something about yourself already? You say something?
No.
What?
Yeah,
Yeah.
I think.
Okay, and then you say about because in the study that you're mentioning on cortisol is the one conducted by who did this work?
By Barthelmess, I don't remember exactly. It was several researchers working into that, some Italian, some American, but I really refer to that in papers 15 years ago.
Yeah. Why don't you say that? I feel that eventually people would come, but we then you introduce yourself, I introduce myself, you say something.
Yeah.
If Fabio does not know you, if you say something about yourself, then.
Yeah. My name is Fabio Pertoli. I am from Brazil, and I did my master degree and part of my PhD with animal breeding. So I am a little bit out of the ethology and this area. So I am advised by Luis Lemon Coutinho in São Paulo University, and now I'm doing a postdoc, postdoctoral, right? And in 2015, I went to Sweden to start with this study with the internet. So the idea was to mix the method that we applied in chickens in the animal breeding contest with the the technique that Carlos and Jensen was working in Sweden, that is the methylation. So we mixed both techniques to develop this new one and worked well. So and now I'm doing postdoc we in partnership with Jensen and Carlos. Unfortunately, he's not here today, but yeah.
Carlos Buzan is from Chile
Yeah.
originally and was at Washington State, and I put him in touch with Per, and he worked on the group with Skinner in the Washington State, one of the real pioneers on epigenetics. And he was supposed to be here with us, but had a last minute type of commitment. And the reason why I brought together, because I do feel that these disciplines are evolving quite separate. And would I say something about?
Yeah, so a lot of my early work was on aspects of early experience on behavior development.
Right.
So that's where I started, and then I got to do everything work. And at that time, there wasn't any epigenetics, but
One of your first publications on that, huh?
my first publications was on early experience using the epigenetics.
Yeah.
So really issues and how behavior change according to that was the experience after acting. So it's quite a long span of interest in neurons and the series of studies on mammals and behavior development in mammals. But this is a long time ago now. And I know very little about this today. So you can teach me that thing. So I haven't been working on that at all, although I have listened to 2 or 3 papers with experiences, given them with relatively things. But so I don't know much about that at all, except that it's anything to it's just amusing myself.
My name is Björn Rolf. I'm working at the University of Life Sciences as a professor of ecology and basic ecology. My main interest is behavior and behavior development. I also do not know very much about epigenetics. But we have done a large number of studies on effects of cancer expressed in different species, like foxes, chickens, salmon, and goats, I think. More I don't know, but at least those mainly. And will also mainly effects of stress in the last third of the pregnancy, not in the HB axis is more or less mature, and effects on different kinds of behavior. And I also wrote the new article about this in 1998. I think I referred to some of the studies there also, if I remember correctly. I have it in the computer. And what we see and also in the review article as well, that things that can affect almost all kinds of behavior. Affect development, affects learning behavior, social behavior, reproductive behavior, and cognitive behavior, cognition. It can affect almost anything, and at least it seems like at that time I think those literature related most of this to the effects on the cortisol receptors in the hippocampus, affecting the amount of or increasing the cortisol production and the also but all that kind of 20 years ago, people wondered if there were some other effects in addition, which was not found yet. So it may not be only about cortisol, and at least the cortisol effects are important in outcome of different behaviors. And it's a long-lasting effect which can go on to the next generation. But really, it is affecting the reproductive and then the affecting the second generation also. That makes it really nice.
I got interested in this area also a long time ago, but probably the first work that I then was postnatal pig that work on aggression with the early weaning that we did in that every National Geographic coming to Norway from, and that was the. And then on the prenatal part, we probably the most exciting bit was the one with Marjorie in Norway when we looked at the changes in the embryonic spine in the animals according to handling stress. And we put together also studies on the goals on the AWIN project, and we had some initial ideas on the development of tail biting pigs related to the prenatal environment, related to this potential inflammatory process, and then then sheep, goats, a little bit of pig postnatal, and now in Brazil we have we've completed a study looking at hunger in piglets. You've reviewed the paper, the hunger in piglets, and the sows with effect in piglets that have been published at increasing aggression. So also the meeting on Thiago presented this.
Yeah.
And she's been looking at environmental enrichment and abnormal behavior in we have a poster where we looked at lameness in sows and the consequence of a sow being lame for the offspring. That's not been published. It was a kind of that's the poster we brought to the ISAE where Marisol looked at a data set that Patricia and Thiago studied and identified that offspring of sows that lameness at a higher level showed more aggression at weaning, and the days after weaning, they also show less pronounced development in terms of weight gain. And that's and lameness is a condition that kind of is present in large number of sows, and we suspect that over 70% of sows are lame. 145. We speak about cows 30%, but sows the number is quite. And
Do you think that inflammatory responses are part of what's going on in those cells that is having the effect?
absolutely, these type of thoughts don't. I've been interested since when I was in Germany.
Yeah.
There's nothing more competent in altering the kind of a cell than well, cortisol does a very good job in causing an effect, but also inflammatory cytokines. But there's so broad, there's so many of them.
I'm
Yeah.
questioning this because so if they have lame cells, then the cell will be feeling some pain. There will be some inflammation going on.
Yeah.
There will be several things
Yeah.
happening. If there is a consequence, when it has a consequence on the piglet, is it something to do with the animal feeling the pain, or is it something to do with inflammation? They're overlapping, but they're not the same thing.
We try to separate that.
Yeah.
That's why in a study that we did with LPS,
Yeah.
that's LPS is this.
It's inflammation without much pain.
Yeah, there's no pain. It's only
No
temporary.
pain.
No pain. In theory, there's only discomfort. It's it's like this gram-negative membrane
Yeah.
from bacteria, and but we could not do that.
Are you sure there's no pain?
I'm not. Well,
Because sometimes if you have enough LPS, it does maybe causes pain. So
And-
I don't know.
it causes massive discomfort. I mean,
Yeah. Because
massive discomfort.
LPS
there is fever. But there is another difference in these two kind of protocols because the
Why don't you put the screen here?
LPS is too fast. The laminar
Thank you, though. Yeah. But then I answer your question. Then
No, she doesn't.
the
Go ahead,
difference
yeah.
in the two protocols because the LPS is too fast. The effects maybe 2 hours, and the limit is during all gestation
Yeah.
in different degrees.
Okay,
but we get-
effective LPS is very brief.
Yeah, yeah.
4 hours, yeah. Yeah. And
Yeah.
we in lambs, in pregnant ewes, we managed to use LPS, and it caused a change in the offspring in sheep. In sows, we could not use LPS because in sows, LPS caused abortion. Then the model of LPS, I call Robert Danza in the US, gave me some other ideas because in sows we cannot use LPS. Then in sows, what we did, we tested in the prior to copulation with the idea that it may change it. And we and the other problem with this inflammatory cytokine is the real issue has to do with how to measure them. How to measure them. I spent when I was in Norway, I spent a huge amount of money setting up protocols to measure cytokines in pigs. I did the same. I did the same in the UK. The lab in the UK with Beatrice Morone worked, and we developed a protocol to measure cytokines in pigs. But they're species specific, meaning that we spend now with Fabio almost 25,000 reais buying kits to measure cytokines, and they're no good. They're no good. Meaning the methodology
Sorry, what's the
kits
the hits on that?
no good from I'm not going to say the name, but the available methodology to measure them. What we did with cytokines, what we can do quite nicely is looking at gene expression in the central nervous system. We've done that in the past, and what we know is that their action is not separated from cortisol because in a sense cortisol some of these cytokines cortisol can reduce because cortisol is anti-inflammatory, and whether with others has no effect. But the interplay and most of the but when people look at that's why, like Fernando, an assessment of the pathology at the site, if we can identify where the problem occurs, but sometimes the effect in the brain leaves absolutely no sequela. There's nothing that you can really measure there. I mean, what you know, there's an effect on the behavior. But to me, the possibility of having a clean model, because you're totally correct. A lame animal probably eat less frequent, it's more fearful, it's going to avoid. Then there's a whole set of things happening around lameness that make as a model a very difficult one.
Even if you don't know which bit of it is causing the effect,
Yeah.
it's still interesting. So the results are still interesting, but it would be very nice to know which one.
And you can
Which is the
see,
effect.
but the results are incredibly exciting. The results of lame cells, if you can check that. I mean, the piglet born from lame cells gain less weight, fight more, and vocalize. I think vocalize less. The ones from in the open field. And this study came as a result of a data set that was generated in a study by Patrice Chaco and Marisol Sarmiento was a student doing an internship who processed this data. And what we have now from these animals are also measure of cortisol in the placenta. And for these animals, we do not have brain tissue, do we? No. But to me, I mean, and for example, I mean, if there is any some ways of looking with your toolbox, these cells of the immune system, they're quite good possibilities because they seem to be even in the case of humans in things like Alzheimer. There's an inflammatory component to that. The problem is that when it starts, there's an inflammatory component to that. There's new data showing that even obesity. I mean, the point is, inflammatory cytokines they are very good in terms of shaping the receptor in the cells or the source of cells. They have this type of capability. That's basically the way that we're kind of to move ahead on that.
Let me just ask you something, like a general question. A few people didn't talk. and that information is therefore being thought about as something negative. On the other hand, information is extremely adaptive and helps
Yeah.
individuals to cope with what is happening. So what is the right balance? And this is just, so there is information which is adaptive and functional, and then there is information which maybe continues for longer and is having negative consequences.
Yeah.
And what makes it go from one to the other is a very interesting thing. It's a very wide range of questions.
Yeah. But the model, the naturally occurring model.
Nobody answers them.
No. But the naturally occurring models of information, though, and Patricia and Fabio, they're incredible. Think about a full life. I mean, a lame cow may be lame for, I don't know, 60, 90 days. A lame sow, meaning that these models, they're so, or birds. I mean, to be honest with you, I'm the bird, I mean, you've started with the corticosterone in the egg experiment, was one of the very first one where you injected, then you look at the responses. I mean, I have no idea. Do you know if there's any cytokines in the eggs? Happen in the eggs, because or in the ovary.
Not in relation to information.
No,
There wouldn't be information.
no.
There could only be communication. I don't know.
Yeah. I don't know. I also don't know. Probably to record it. We don't know. But the egg model. I don't know the egg as a model. I mean, I think it brings a lot of in many ways. Of course, studies becomes early, but the point is the
bad model for the complex system.
yeah. But in a sense, I mean, if you look at the breeders, the breeding animals, I mean, quite often they may have lesions. And in a sense, I mean, it's likely that but again, in birds, the whole what we call cytokinase in the mammalian species is totally different in birds. I mean, that's what makes the system so complex to study. But to me, I mean, there's a lot of work that could be explored this type of interface, especially if it affect central nervous system, and we do have we need to talk more with people that are kind of course in theory, I mean, if you have people that are more knowledgeable about the about the area of cytokine. We know a bit about different things, but not enough about cytokine. I spoke a fair amount with Robert Danzer. But Danzer, I mean, I talked to Robert of the phone. Yeah, he's doing fine.
There was this morning a reference to Robert, and I said, well, I haven't seen Robert for 6-7 years.
I saw him in France briefly. I think last year, and then I organized to call him because he works now with in the United States, with Houston, Texas, working with cancer patients, and his focus is on the way that his chemotherapy drugs alter the wellness of the patient and kind of one of the issues as a result of these changes in these cytokines.
Yeah.
And he, yeah, I kind of maybe should have a follow-up. Maybe should write down a note to have a follow-up meeting with Danzer. He offered to join us in a specific workshop on cytokines and Pre-Natal program. He was also very concerned with these measures to assess cytokine. And also this model that we use, the LPS model, he had some reservations.
Yeah, what does he say about it?
He had some reservations.
He's quite negative about it.
Yeah, he did use a lot in the past.
Yeah, but he changed it.
Yeah. But he's being basically, and in some of his work now, you see that people people go to this chemotherapy to treat cancer and they feel really, really awful in this process.
Yeah.
And in his data showed that it's cytokines are the ones who basically, which play some role in that.
There was a mention this morning, not so much of cancer patients, but of people recovering from heart attack. And in some forms of treatment of heart attack, the people are much more subject to depression afterwards than with other sorts of treatment. And that was thought to be associated with the difference in cytokines caused by the treatment. I don't remember the detail, even though it was only 2 hours ago.
Well, I know, but this whole discussion on the cytokines, on tail biting,
Yeah.
I introduced that when because I coordinate this project they're discussing right now.
Yeah.
I was me, Linda Keeling, Ana Valros, and to measure the way they measure cytokine is done in the lab that I put together in Edinburgh.
Sure.
There was something from that. Then basically, I'm the one who migrate and end up losing this whole. But the cytokine area it's phenomenally exciting. I'm not sure how much progress they've done or the established the link between cytokine and the thereby. I've not watched
Okay,
it.
I can tell you. There is established problem. Yeah.
Okay, well, they should maybe acknowledge at the end of the paper on generous idea the 1,000. That's brilliant.
The best thing is that when you have an idea and somebody actually uses it instead of ignoring it, then it's good.
Yeah, I think it's very good. Yeah, but it is. I mean, that's very nice. That was in my original research council grant. I mean, it's brilliant. Makes a lot of sense. I got also actually nervous about it. That's all very good. And yeah, you mentioned the issue of depression. Absolutely, depression, depression and cytokines have this tremendous role in changing trajectory of cells. Very nice.
It's not surprising, but what it means is that there are 2 or 3 ways in which the sort of topics that we're talking about can be related to human medicine. And there's no doubt that you're more likely to get some money if you can relate it to human medicine. So we have to put one health, one welfare.
Yeah. Oh, by the way, I have it's nice that the 2 of you are here. I've been invited to organize a workshop at the Brazilian Veterinary Congress, going to be in the end of this year, in December, in Pará, in Belém. And I'm a suggestion for the team is one welfare, one health, one welfare.
Yeah.
We talked about.
Good. Great to do that.
Yeah. So,
I'm going to the World Veterinary Congress at the end of this month
okay.
in China, in Korea.
Yeah.
And I will say something there about welfare.
Yeah. There's a real growing body of work of linking human welfare and animal welfare. That's quite exciting. But back to the
Looking at immune system function, control of it, the role of cytokines is one of the areas where it's very easy to see.
Yeah.
You can put the human work and the non-human work closely together and find out which are relevant to both. And yes, it's just the general idea that good welfare means better immune system function is something which is still not thought of enough in human medicine.
Yeah, and because back again, I mean, when we measure a kind of antibody production stuff is too simple. I mean, it has to do with teaching the immune system, the teaching of the immune system to be even getting them more or less prone to develop a disease such as cancer. Then I mean, I think there's a lot of potential development. Then the first question that I asked is the current toolbox adequate to assess consequences for the environment. I mean, I put a motion that no. I mean, I don't think we have. Would you agree on that? I mean, for both. I mean, for both the behavioral I think we are operating on a
It's good enough to do something useful.
use, but not no
So many
no. And
people will we, if you look back now, 20 years and 40 years, and you see what can be done now and what could not be done 20 years and 40 years ago, then you can see that in 20 years' time people will ask the same question and they will have better tools.
yeah.
But that doesn't mean you shouldn't use the tools we have now and use them.
And then
Doing research rather than nothing.
yeah, correct. And the second thing that you put is I forgot kind of second question. If you think about the framework to develop a collaborative project on prenatal programming, is that a possibility or is too broad? I mean, or if that is the case, then I have a kind of I put some ideas in terms of that. In a sense, I mean that's something that is happening everywhere. I mean, the question is there anything that we can do collaboratively in terms of the development of models for the study of prenatal programming? And I put, let's say, male and female, because I mean, we do very little on males, almost nothing, really. And in terms of,
Males are very important.
I know that. I couldn't agree more than that. Absolutely. You ask Marcia, she probably could yeah, she could make it. But the point is, when it comes to, of course, it's not even prenatal, it's pre-copulation type of programming, like germ cell programming. The difference of male and female is that in the case of female, the template for the germ cells, the foundation is laid during time when they're neutral, and after birth, basically, they carry that. I mean, in the case of males, the germ cells are in theory being exposed to environmental things and have the possibility to adjust. And then, in a sense, I mean, we probably, by thinking about males, we may find a way why they still exist, given the fact that you even said how unimportant they are, but they seem.
It's a joke.
I know.
No, it's not great. No, it's not completely joke. But so, you know, I think
But they carry much less
females
cost.
are more important, but there are lots of possible ways in which experiences in males could have a consequence.
Exactly, yeah.
And that if you look at all the mechanisms for modifying gene expression,
Silence
most
gene,
of
yeah.
them are available for males and not just for females.
Exactly.
So there is plenty of potential there, and that has been ignored.
Perfect.
Because people didn't think it was important. So.
And yeah, and in fact, Jack is for, you cannot speak, right?
Is Jana there?
Jack, yeah.
Oh.
Yeah.
You mean he's
Yeah.
Hi, Jana.
Jack Bernardin is a PhD student in our program, and we asked him questions about males. I mean, Thiago is going to start an experiment getting males into difficult situations and not so difficult situations. And he's going to be looking at genes that are problem solving. That's something that the whole methodology has been kind of discussing. And it's an area that Carlos has some expertise on sperm. He's going to. Yes.
Prior to
Yeah, population. Yes.
population.
Yes. We are doing as we speak. Yeah. And using similar models, using LPS and using environmental enrichment on males, and we also have access to males that naturally are in really difficult conditions, and the idea of are you familiar with the way that they keep male pigs in breeding units down in Bjorn and Fabio?
Yeah, they have terrible time.
Terrible time,
Yeah.
really.
Yeah.
They keep like in
Why?
in
Because
cells.
everybody knows they're unimportant.
Yeah,
That's why.
Yeah.
That's the main problem in farm animal husbandry, that there are too much focuses on females, not on males.
Yeah.
There should be more focus on male welfare.
Yeah.
So when the welfare, you look at boards, bulls, actually rams don't do too bad.
No.
How about male turkeys? Would you like to be a male turkey? They can't breed at all, they can't populate.
No.
Anyway, these animals they are treated in a quite different way, which on the whole is worse than the way the females are treated. So what's the consequence of that? You don't question.
Yeah, and that's a question that, in a sense, I mean, if I would be in a position to make a decision with a group in terms of this large project, I'd like to bring two lines of thoughts together. I mean, female and male. In a sense, I mean, it could be that if the conditions are really difficult for a female, there's nothing in the sperm of the male that can fix it. But if both of them are going to the condition of having this hard time, well, chances of things getting better are really limited. And and for us also, Bjorn and Don, we are getting interested in this period that, let's say, in South, that are prior to copulation, or in the beginning of the copulation, that is also being disregarded as an important, of course not in Norway, but in Europe you can still keep pregnant sows in gestation period up to the 4 week after mating.
It has been some studies on chickens time ago about the pre-natal stress.
Oh,
Yeah.
chickens.
So they studied that. But we don't remember name, but that's what I found in my review article from 1998.
Okay.
Because they're looking particularly then on the environmental situation during the mating situation, and found some effects on the offspring. So not only prenatal and pre-mating stress
Pre-mating.
can affect offspring.
Right.
But there are not one on chickens. Yeah,
Yeah.
it's on my paper and
Very, very good. Yeah.
But
And recently, I think Daniel Matt on Perry Jensen's group did some of the work on the fasted chickens also on pre-mating and the way that capacity.
So that's one reason.
Yeah, and the focus on males is something relatively important for the issue of genetic, and in terms of bigger area for research, we have nutrition is probably one of which got the most attention so far. Prenatal nutrition during pregnancy of different species, including humans. I mean, omega-3 fat content, and in our case, fiber. Then nutrition is a route that has been nicely established. Yeah, meaning that is an area that, yeah, meaning it's an area that I think our impact is going to be limited, given the fact of the type of work that's being carried out. I don't see us being able to make real contribution in terms of nutrition, given that's what I'm trying to say. Yeah.
Well, I'm going to say, you said, in terms of what are the questions to ask and what are the studies to actually include?
Correct.
The difficulty is to narrow it down to what studies are the ones which are really well.
Exactly.
But you absolutely have to do that, and you hope that you will answer all the questions. You can say, well, we may get something about these areas may get something useful, but the main thing is to think what can actually be done,
Yeah.
and then what are the consequences which could be realistically results that could realistically be obtained, and therefore the areas where we could be likely to have some
Success.
some success.
Yeah. listed.
the way to do it.
Yeah, I listed as a number one. Of course, my favorite for many years is this issue of diseases that are associated with like protection diseases or diseases associated with inflammatory processes. And then in our specific case, I mean, lameness or locomotor disease is something that I'm quite keen as being part of the of the model. The Of course, the other area that I come back to the disease later on with some ideas, see if we can come up with a clean type of.
If you do that, then you're going to have a whole set of control situations.
Correct.
The diseases have such wide range of effect.
Correct. Yeah.
I'm not saying don't do it. I think you need to be able to have other things that you're actually trying to see that are the effect.
Correct.
Cause of them.
Correct.
Diseases have several.
I know. I put here social and physical environment, but again, it's a bit of complex. This is something that is of interest to.
How many people are going to be involved in the project? You don't know the answer.
From Brazil, we have 3 groups, and UK probably 2 or 3 groups.
Yeah. So you might have 6 people.
Yes.
Okay. Because one of them which is realistic for 6 people.
And we what in a sense the ideal. Yeah. If it gets into this in this area of BBSRC, we need to be careful because we wrote a grant 2 years ago on the same project. We've been criticized by asking too much money. But
6 people is a very big project.
yeah.
I mean, most of the time when I was trying to get funds, I was looking at only one person, and you couldn't get money for 2. So, this is the difficulty. If you're talking about, say, going to the and this is because the UK funding, the way it works, that's the way they tend to do it.
Mm-hmm.
If it's an EU project and you've got more collaborators, then that's a different situation. Or, I don't know how Bethesda is working in terms of this kind of collaboration. But, so I think you start by saying what is a realistic amount of money to ask
Right.
for, and then you say how many people could we have, and this will mean there are several collaborators, and then that's going to limit what you actually write in the application.
Right. I mean, I think the limit is going to be in terms of which animal species are you going to be studying. I'd love to have a human component of that, meaning that if a disease process will be selected, we need to have some type of similarities. And did you find out, Björn?
About the pre-natal, I remember long as the studies were going on rats by different roles and ordered from the doctor in 1965. Then they found that if it was in impoverished conditions prior to mating and similar conditions after mating, then the rat pups which had been conceived with the impoverished condition before mating, they spent less time in the same development in higher settings. And there was some from conditioning also.
More care is impoverished if the prenatal condition is
Yeah. And then the conditions after mating were the same.
That's really exciting. In fact, we have now 2 studies being carried out during the pre-mating. One, that will help with pigs. We are looking at ewes, and the last we synchronized the animals, and was 5 days before the expected estrus. We've we've challenged the animals with the LPS, and some animals we put in crates in some type of groups, and we monitor the biomarkers of stress, and we also look at the microbiota into the vagina of the animals during this period, and then after a week, 5 days after estrus, we put the animals down and we collected ovaries, uterus, vagina, and bladder, and we've looked at microbiota, the bacteriology, and we are now studying genome of the bacteria on that, with the idea that that's another component that we rarely speak, but with these inflammatory responses, there are changes also on the presence of a few bacteria that also have quite a powerful way of making some of these changes. Then we're looking at the prenatal and back to the ovaries, and uterus, and the data on the microbiology is all done, and the genomic of bacteria in the process of completed. With the idea that no, it's being just
will come later.
yeah, hope it come yeah, but it's the
You can't refer to it now.
no, we're not, no. But what I think what we can say, Patricia, help the experiment that having the animals into the three type of environment. Can you say how they respond to LPS, Patricia? How they respond to the LPS?
The kids that were in the
Paddocks, paddocks.
picket. It was really good after 1 hour, and walking and nosing the floor, the straw, and the youths that it was in crates. It was really bad. Lying down and
Right.
The same challenge induced very different,
yeah.
very different behavioral responses to LPS. Oh, here things cut to white. It's quite rare now. No, it's not. It's not. Yeah, I kind of apologize for these.
Is somebody a bit fatter?
Yeah, that's right. Never tell them that. Never tell her that. Never tell her that. I haven't seen her for 3 years. For you. I make assumption that she behaves like me. I accumulated more kids than I. Apologies. What is Janela? Let's open the window.
I may look at this. There will be the other group has coffee available.
Right?
Maybe about 3:30 there should be coffee there.
Yeah, we go there and we
It's not perfect coffee, but it's coffee.
okay. No, and
Not perfect music.
was brought into the discussion here also the issue that like the technologies for assisted reproduction. Brazil uses tremendously, let's say, in vitro fertilization in animal production. We use that for beef cattle a lot. And Fabio was bringing this issue to the discussion. I mean, whether or not, what are the consequences for that in terms of epigenetic type of, and I put it as an added. The only problem is in terms of turkeys. Yeah, but in the beef industry, it's used a lot, and you can say something about it.
Yes, I hear about that in cows and swine that the mortality is bigger when you have the fertilization in vitro, right? So of course the environment is totally different when we fertilize in vitro and when it's in vivo, right? So we can look for epigenetic changes in the different conditions. I was talking with Ricardo because his wife works with the
Yeah,
swine, and maybe she can get some samples for him to try to analyze the epigenetic difference between these conditions. It's really interesting. So, but the issue um, is when you get this embryo, because you have to get in the early stage of the as soon as you fertilize, you have to get, because as soon as you start to divide the cells, so it's more complicated because you have a different tissues, different genes expressing
Right.
and different. So as soon as we plan how we will do this,
right.
maybe in the beginning of the development,
Right.
and he told me that they slaughter a lot of swines and they can get in vivo different
Yeah.
stages,
Yeah.
and yeah, I think it's a really interesting experiment to do and to analyze
In the
it.
early years of the in vitro fertilization, they used to get animals that are much larger than the naturally. But these they managed to change. They understood. I don't remember exactly what it was mechanism that generate much larger offspring. And but is there, anything else? Is there anything else in terms of areas? Then I put here then disease, nutrition, social visual environment, reproductive technology. And in the order reflects my real preference. I mean, disease is, of course, when it comes to the diseases, I mean, it would be really useful also to narrow down. And for us in Brazil now, we have a lot of concern with Zika, chikungunya, and dengue.
I think that in deciding what to do, one question is what can be done, and another question is what are the issues which are really important right now.
Okay.
And obviously that is really important right now in human medicine. So, yeah, so it's sensible to think about both of these things.
Okay. And in
But what I mean, are you thinking of presenting it as a model for Zika?
uh, uh,
And if so, what would be a model?
that's I've thought about that, and the truth is that none of these viruses are tested, and I think it's probably not very safe to adapt to
Well,
a
no, I can, and you're pregnant as well.
yeah.
Probably you are. So you can do it.
I could do that, yeah. I could do that. But but to me more in terms of not demonstrate the phenomenon itself. Uh, our goal in our team is to find ways that we could really mitigate. And when I say mitigate, let's see if we identify that the process that creates this type of changes, the process is related to some dysfunctional release of one of the cytokines, I don't know, IO10.
Yeah.
Then in theory, you could uh, uh, have an antibody against IO10 and deplete the body from IO10, or you could have some type of antagonist to IO10. In a sense, I mean, what I'm most keen is on the possibility of mitigating this effect. I mean, demonstrating the effect itself is of course the first part of the process, but the situation right now, especially with Zika and chikungunya, has to do with this microcephaly, this poor development of the brain. But again, but this type of disease, the model that we know, they are not very useful because what has been demonstrated is that the virus already in fact, really penetrate the central nervous system. It's not an impact like we discussed of, even though dengue, the truth is, but the fever state, the fever state that dengue created, uh, uh, um, no one really knows what that may have impacted and we, and for that, we have even a possibility, the human component of the study could be even an epidemiological study, even an epidemiological study of pregnant women that develop that dengue. With Zika and chikungunya is a different business because we know the kind of impact. But if dengue in febrile state of dengue uh, 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 creates some type of impact on the development of kids, then our models are relatively useful because the issue with dengue is fever.
Nobody has demonstrated that at the moment, is that right?
No, I haven't.
Anybody got?
No, no.
No?
I never came across. We could, maybe that's a task. Let's write down a task first. Check for dengue.
Do a literature search for that one.
Yeah.
Because I wouldn't know whether it's been done.
And
Is there a
database.
yeah, so first of all, the investigation, if you would so that you don't say something which is not.
Right.
Because and then what would the you're not going to use baby, what are you going to use? What's the model? What disease? In what species? What the you want something which causes a fever?
Yeah. Well, I
Which
mean,
one?
at the moment, the only one that I've tried is LPS, but I don't know how.
That's not a disease.
No.
That's a control. That's a further investigation.
Yeah. And I don't think there's any references in. We could look into that. Yeah. Dengue in pigs. I haven't seen that. Yeah. Then I don't know. No, I don't know. And
Interesting. I think that's a really interesting suggestion. That what happens if you have a fever during pregnancy?
yeah,
That's an interesting thing. And it obviously will be possible to do that with the pathogen and with another.
yeah. And this is something that
And it would be one of the
yeah, it would be very, very relevant.
And we'll have some general, it would be possible to have some general
Correct.
theoretical consequences of finding that out.
Correct. Right. And shall we go and get their 3:30 their coffee?
I think that probably there will be, the payments were there just now. I think they will be putting new ones again because they said there's going to be a coffee break in the afternoon.
Shall we get the coffee if not there, and then we see if our colleagues would appear? Because to me, there is in terms of I'm enjoying the discussion a lot, and the one of the of course the stretches, I mean, really far apart, but hi. The feedback is here. Patricia,
Hello.
How are you? Good to see you. How are you? I'm fine. Hello. Aline.
and Bjorn Brastad from Norway. You're just going to start talking about sheep and sheep immortality of plants.
Yeah.
Why don't you go and let's get a quick coffee, then we come back here. Let's get just before you go, then why don't you say something? Patricia, introduce yourself just for then you go the coffee know each other.
Okay, I'm Patricia, I'm PhD student. I'm working with environmental enrichment in the south, in the outcomes in the offspring. Yeah.
I am Fabio Persil. I'm from University of São Paulo, advised by Luiz Lemon Coutinho. and I work with epigenetics and bioinformatics related nowadays with behavior and this, but I work at a lot with animal breeding, and so now I work more with well-being.
You to know them, you don't know Bjorn, huh?
No, I don't. I think we
Yeah, maybe I'm from the University of Norwegian University of Life Sciences. I've earlier been working on different species, but not the last 15 years.
And you can then you go for coffee, and you and Aline, he's been in the area for quite a while. Would you like to say something about you? Diz alguma coisa pra
Who are
você?
you?
Who are you? Hello.
I'm Paulo from the Uruguay University of Pública, working mainly in dominant reproduction, behavior, and endocrinology, sheep, and dogs, and something happening here.
And I work with Uruguay and Italy. I work mainly sheep behavior in new land form. And I'm ready.
Let's have a coffee, and then we come back, and maybe can. Avisa o pessoal que não tem copo virtual.
I think we're